Hi, my name is Linda Haltiner. I work at EWAC and I will give you a short insight into part of my PhD research. Today I will talk about life in the deep, colonization by Dreissena along a depth gradient. Due to their high filtration rates, high abundances, quick spread within and between the lakes, and the ability to colonize various substrates, quagga mussels are challenging our existing lake ecosystems. Quagga mussels appear in high abundances as seen here on this pipe on the shore of Lake Geneva. Adult quagga mussels were first discovered in Switzerland almost 5 years ago in Lake Geneva and Lake Constance. Now they already spread to other lakes in the western part of Switzerland. When quagga mussels colonize a lake, they are able to colonize the whole shore within a few years, as seen in Lake Constance on the left. On the right in Lake Michigan, we see that within 10 years since the first discovery on the shoreline, they colonized also the deepest parts of the lake. Until now in Europe, most quagga mussel monitorings have only looked in the littoral zone, but we know from North America, where quagga mussels invaded more than 30 years ago, that quagga mussel can live deep. And that's what I want to talk about today. We are running experiments in Lake Constance and Lake Geneva that examine how deep quagga mussels can be found on introduced substrates and whether there is evidence that they are adapted to the depths where they are found. To answer those questions, we have installed a settlement experiment. It's a rope extending from the surface to almost the lake floor, over 100 meters long. On this rope, we attach settlement plates, and every 3 months, we check the colonization of quagga mussels on the plates. The experiment is located in Lake Constance in the Überlinger See, attached to the railing of a EU buoy of the University in Constance. The second location of the experiment is in Lake Geneva, in the perimeter of the Explore platform, attached to the orange buoys you can see here. The experiment is still ongoing and will continue in 2021, but I can tell you that the mussels appear to settle faster in the upper water column. Mussels grow all over the shallower plates. In the deeper areas, settlement seems slower, even though adults can be found in the deepest part of the lakes. On the same rope as the settlement experiment, we perform a reciprocal transplant experiment of mussels collected in shallow and deep areas. We see differences in morphology between deep and shallow living mussels. Shallow mussels appear dark and robust, while mussels from the deep are lighter and more fragile. And we place them in bags and then hang them on the ropes in both depths to see if there is evidence of adaptation to living in the deep. Reciprocal transplant experiments are very powerful tools for understanding adaptation. By moving individuals between environments, we can look at how survival changes in their home and transplanted environments. Home environment means mussels from deep areas are placed to live also in the deep, while transplant environments are mussels from the deep forced to live in the shallow area of a lake. And the same for mussels from a shallow origin. If the survival is higher in the home environment and worse in the transplanted environment, then there is evidence that they are adapted to the home environment, in this case to depth where they are found. This experiment is also still ongoing and numbers are still prone to change. But in the first results after 3 weeks of the experiment, we do not see the expected adaptation pattern. Irrespective of where the mussels were collected from, they survived better in the deep. I am curious how and if the pattern will change the longer the experiment lasts. These experiments have been made possible by the infrastructure provided in Lake Geneva by the L'expert platform and Lake Constance by the EU-funded instrument buoy, and the funding from Seewandel. And without technical help, no rope would be floating in the lake. So a great thanks to Martin Wolf and Alfred Zulger from Uni Constance, Sébastien Lavanchi from EPFL and Michi Plus from EWAC at the L'explore platform, Rafi Bossart and Amruta Rajarajan, and especially Silvana Käser and Steff Walcher from EWAC. Thank you.